Hello, everyone. My name is Reema Alkhalifah. I'm a second-year medical student. Here's my contact information. If you have any questions, feel free to contact me after the lecture. For today, I'm going to be talking about glomerular filtration and renal plasma flow, both parts. So it's your third and fourth physiology lectures. For today, I'm going to talk about the filtration membrane, its anatomical structure, and the factors in it that affect filtration. So by the end of this lecture, you will be able to understand them, inshallah, and recognize what factors affect what. I'm also going to briefly talk about glomerular diseases, but I'm going to go through it quickly because Dr. Shams emphasized that he will not be asking any questions. But still, it's good to know how these diseases work. I'm going to also talk about GFR. So you're going to be able to identify what affects it. And finally, I'm going to talk about how we regulate renal blood flow. And there's two types. There's autoregulation and there's the other mechanisms that we'll get into at the end of the lecture. طيب? Okay, bismillah. So before we begin, let's talk about basic anatomy. So we know this is the kidney. It has multiple functions, including the ability to excrete metabolic waste products. And if we zoom in, we're going to find its functional unit, which is the nephron. Now, the nephron has two components, the renal corpuscle over here and the tubular system. As you can see, it's all twisted. It's not always straight. And then these are the other vessels, including the capillaries, the peritubular capillaries. But for today, we're just going to be focusing on the renal corpuscle, which is over here if we zoom in a bit more. And we're going to talk about its structures. Now, this is the afferent arteriole. This is the efferent arteriole. So you can notice the blood is going to go into these capillaries. This is a capillary bed, which is called the glomerulus. And this outside structure is called the Bowman's capsule. Now, the Bowman's capsule has two layers, the parietal and outside layer, which is this one over here, and then the visceral layer, which is the podocytes, which are also specialized epithelial cells. Now, the podocytes surround the glomerulus, so it's part of the Bowman's capsule, but it also is associated with the glomerulus by affecting the filtration of the plasma. And let's say we're going to cut a cross-section picture over here. If we cut a cross-sectional image is going to appear. Now, this is the Bowman's capsule. This is the outside layer. This is the Bowman's space over here. This is the Bowman's space. And this is the filtration membrane, which is the glomerular capillary. Now, there are three important layers in the filtration membrane that we need to get into. From inwards to outwards, we have the capillary endothelium, which is right next to the lumen of the capillary. Then we got the basement membrane, and finally the podocytes, which form the outer layer. Now, this is very, very important for filtration. Each layer contributes to a certain factor. If you have any questions, feel free to type in the chat. I'm going to be able to read them, inshallah, and we'll answer any questions you have. So, now we go back to the filtration membrane. Again, from inwards to outwards, we have the capillary endothelium, the basement membrane, and the podocytes. Now, what can it filter? What can it freely filter? It can freely filter water, solutes like sodium, urea, glucose, and others. And it can also filter small proteins and peptides. But what it cannot filter is larger proteins, especially anionic proteins, red blood cells, white blood cells, and platelets. Now, this is the blood. It gets filtered out, and once it's in the Bowman space, it's called an ultrafiltrate. And we're going to get into how they affect filtration. So, first things first, capillary endothelium has pores that range from 50 to 100 nanometers, and it prevents the filtration of cells such as red blood cells. But it can still help with, like the plasma proteins can still pass through the endothelium, but it specifically does not allow cells to pass through this layer. And they also express negatively charged glycoproteins. And with the addition of having its role as a filtration like barrier, it also synthesizes vasoactive substances like nitric oxide, which is a vasodilator, and endothelin 1, which is a vasoconstrictor. Now, those are important for controlling the renal plasma flow. And secondly, we have the basement membrane. Now, here it contains polyanionic glycoproteins, which allows it to be negatively charged. And now here, this is where plasma proteins are not allowed to enter. And keep in mind, basement membrane is the main or dominant layer that is charge selective. And finally, we have the podocytes. Again, they have slots that are ranging from 20 to 50 nanometers. It's filled with polysaccharides. Now, the sialoprotein is the glycoprotein that has polysaccharides as its component. And the podocytes also have a thin coat, which is the glycocalyx. Now, the glycocalyx, the sialoprotein, the glycoproteins, all of this contribute to the negatively charged filtration membrane, and we'll see how this is important later on. Okay, so these are images. Here we are in the capillary. You can see those holes; those are the pores of the endothelium. And if we look from the outside, now we're in the Bowman space. This is the podocyte, and this is the foot processes that surround the glomerular capillaries. Okay. Now we're going to get into what affects the filtration of these substances. So first, we're going to look into the size. If the substances have a molecular radius of less than 18, they are freely filtered. If it ranges from 18 to 42, they're partially filtered. And if they're greater than 42, this is where they're not filtered through the membrane. Now, we're going to look into this second category. If the substance ranges from 18 to 42, now we're going to look at their charge. So, we said that the membrane is negatively charged. And from past knowledge, we know that charges that are alike, they repel, and charges that are different, they attract. So, if the membrane is negatively charged, what charge do you expect will be repelled? The negatively charged substances. So, it's going to make it harder for them to be filtered through the membrane. Now, let's put this into the graph. This is a neutral substance. Now, notice this graph is only from 18 to 42 molecular radius. Now, let's take 26, for example. This is the negatively charged substance, and this is the positively charged substance. What do you notice? You notice that the positively charged substances filter more than the negatively charged substances. This is at around 0.1 filtrability, and this is 0.9. So keep that in mind. Now, this is just to confirm your understanding. Now look at the molecular radius. This is all less than 18. What you can notice is they're freely filtered. Now, this is 0.98, but it's still considered nearly being filtered. And this is where the radius gets larger than 18, and it ranges from 18 to 42. As you can notice, as it gets larger, the filtrability decreases. Okay? Now, we talked about the 18 to 42 molecular radius. Now we're going to talk about Donan distribution. What does it state? It states that for particles that are smaller than 18, the negatively charged ions are filtered more than the positively charged ions, which is completely different from what we've seen in the 18 to 42 range. Now, why is that? Let's look at this image. This is sodium, positively charged. This is chlorine, which is negatively charged. Chloride. Now, this is a plasma protein. The plasma proteins are usually negatively charged. So let's take albumin as an example. Plasma proteins, as I mentioned previously, they're not filtered through the membrane, so they're stuck in the plasma. And we said that different charges attract. So the negatively charged albumin is going to attract the sodium. which makes it harder to be filtered. And unlike for chloride, it's negatively charged, so it repels it and it moves through the membrane. Now, this is only applied for freely filtered substances, which are less than 18. Now, you're going to say, since it's freely filtered, why isn't all the substance filtered through the membrane? This rule is only applied for the effect of the plasma proteins. It doesn't have any trouble moving through the membrane itself because, again, it's small, so it doesn't have any contact with the walls of the pores, for instance, to be affected by the charge of the membrane itself. It's morely affected with the protein itself. And so here's a simple rule. So this is the sodium concentration in the ultrafiltrate over the plasma. In the ultrafiltrate, it's slightly less than the plasma. It's 95%. Same thing for chloride. It's negatively charged, so more is going into the ultrafiltrate, and R is just a coefficient to help you understand the application of the Donan distribution. Okay, so this number, this 5%, is very small, so it doesn't really affect how much passes through the ultrafiltrate, but still, plasma proteins have an effect to an extent to the ions that are planning to pass through the membrane, and those are the freely filtered ones that are less than 18. So any more questions before we answer this question? Okay, let's type in the chat what answer you think it is. The physician uses the image of the renal glomerular filtration apparatus in the image to explain to a patient his proteinuria. So there is protein that is present in the urine. So which label, A, B, or C, is the primary barrier that normally inhibits the passage of albumin from the capillary into the Bowman space? Okay. Yes, correct. It's B. So it's asked for the primary barrier. Now, what did we say previously? That the basement membrane is the dominant barrier that helps select what particles pass through depending on their charge. Now, the endothelium and the podocytes do help to an extent with the charge selective process, but however, they're more on the size selective side. Now, here's another question. A substance with a molecular radius of 30 and has a strong negative charge is filtered at a lower rate than expected. Which part of the filtration membrane is primarily responsible? Does anyone have any answer? Okay, so the answer is D. Negatively charged basement membrane. So we're applying the same concept as the previous question. A and B, they're more size selective, and specifically choice D specified that it's negatively charged, and this is what is emphasized. This is what primarily allows the selectivity of negative or positive charges along the membrane. Okay, so we're done. Now we're going to move on the diseases of the glomerulus. Again, you're not going to be tested on this. I'm going to go through it very fast. But it's really good to know and apply the concepts that you've learned on diseases, okay? So we've got two main categories. We've got nephritic and nephrotic diseases. Now, keep in mind the differences between them with the layer that it affects in the membrane and its pathology. Now, nephritic disease is inflammatory. So it affects endothelial cells. They get inflamed, lumen gets narrower, and you can see how the lumen, you can barely see any space because of the capillaries. They're so inflamed. And this compromises the blood flow and filtration. So GFR and renal plasma flow decreases. Now, there is going to be mild protein loss, so less albumin in the blood. There's going to be red and white cells in urine. Why? Because endothelial cells help prevent the filtration of cells. So once it's damaged, red and white cells can pass through and go into the urine, and it will give us hematuria, which is the discoloration of urine, dark brown to red. and GFR is decreased because the capillary radius is decreased. There's going to be more nitrogen in the urine because, again, filtration membrane is being compromised right now. And because GFR is decreased, there's less urine volume, and there's hypertension and mild edema. We're going to get into how GFR affects hypertension at the end of the lecture. And mild edema is caused by protein loss in the urine. Nephrotic disease is non-inflammatory, and it's usually secondary to diabetes or idiopathic. Idiopathic means the cause is unknown. Now, because it's non-inflammatory, you can still see there's lumen here, okay? And this disease affects the podocytes, unlike nephrotic disease where it affects the endothelial cells. And here, the basement membrane is thickened and the filtration slits are damaged. Now, podocytes help prevent plasma proteins from passing through. So once it's damaged, there's a huge protein loss. And there's also severe edema because, again, edema is caused by protein loss. So if there's a severe protein loss, there's also severe edema. And there's no change in GFR, unlike nephrotic disease. And the urine volume is normal, but it's foamy because there's more protein than normal in the urine. This is a table just summarizing quick facts. Now, the layer affected in the nephrotic disease is endothelial cells. In nephrotic disease, it's podocytes. Now, in nephrotic disease, you'll notice hematuria. Nephrotic disease, you'll notice massive proteinuria. And nephrotic disease usually affects the magnitude of the filtration because, again, we said GFR is reduced. Nephrotic disease affects the composition of the filtration because we said more proteins are being lost in the urine. Okay. Now we're done with glomerular diseases. Those are properties of human kidney. Each kidney is weighted with 150 grams. Since we have 2 kidneys, we have 300 grams. There's 2.5 million nephrons in the kidneys. The total area is 3 meters squared, cubic squared, and the total area of the pores is 0.15, which is nearly 5% of the whole area of the glomerular capillaries. And this is why there's so many filtration. So, moving on to filtration rates. It's how much of a substance is being filtered. It depends on the plasma concentration of the substance and the GFR. The normal range of the GFR is usually 175 liters per day, okay? So if you just multiply the concentration of sodium, for instance, with 175, this is what it's given. This is the number that you're going to find. Now, this is only used for freely filtered substances, not partially filtered substances, okay? And you can notice here, a high percentage of what is being filtered is going to be reabsorbed in the tubular system. So it's a huge number. It's more than 99% for these substances. Okay. Now we're going to move into GFR. GFR is how much of the fluid is being filtered per unit of time. And it's mainly influenced by the filtration constant, which is the permeability and the surface area of the glomerular capillary. So obviously, if it's more permeable or has a higher surface area, the filtration constant is going to be higher, ultimately making GFR higher. And Starling forces also affect GFR, which we're going to be going into in a few minutes. Now, those are not the only ones that affect GFR. There's so many other factors that influence it, including what is related to the patient. So the patient's age, sex, weight, height, if there's any medication the patient is taking, if the patient is suffering from any diseases, all of these affect GFR. Now, before I get into glomerular capillaries, I'm going to quickly talk about systemic capillaries, but they're not important for your exam. It's just going to be there for the sake of comparison with the glomerular capillaries, okay? So, now those are the Starling forces, 4 forces. It's divided into the hydrostatic forces and the oncotic or colloid osmotic pressure. It has two names. You can call it either way. Now the hydrostatic pressures, they push fluid out. So hydrostatic capillary pressure pushes the fluid out into the interstitial fluid. The hydrostatic interstitial fluid pushes fluid back into the capillary. This is not the case with oncotic pressures. Oncotic pressures pull the fluid, okay? So capillary oncotic pressure, it prefers pulling the fluid back into the capillary. The interstitial fluid, oncotic pressure, helps pull fluid into the interstitial fluid. Now, in the systemic capillaries, only 0.5 of the renal blood flow is being filtered from the capillary. This is a very, very tiny amount. And this affects the changes that happen in these Starling forces. Now, let's talk about this hydrostatic capillary pressure. Now, it's going to decrease as it goes along the capillary. Why? Because there's resistance. And if there's resistance, it's going to affect the pressure. So there's going to be a pressure drop as it goes along the capillary. But for the oncotic capillary pressure, it's going to only slightly rise, just a little bit. So we're going to ignore and assume it stays the same throughout the capillary. For the interstitial fluid, it stays the same. Why? Because when the fluid is being filtered, it's not going to stay in the interstitial fluid. It's going to continue into the lymph. So the fluid here stays relatively constant. Now, finally, the oncotic interstitial fluid, it's going to stay the same. Why? Because we know plasma proteins aren't being filtered in the capillary. Barely any gets filtered. So the volume here stays constant. Now, this is the equation. As you can see, the difference between the hydrostatic pressure minus the difference between the oncotic pressures. So, when the oncotic pressures, the difference between them is higher than the difference between the hydrostatic pressures, this is where reabsorption is favored. When the hydrostatic pressures are, the differences is more than the oncotic difference, it's going to filter the fluid. Now, so the arterial end of the capillary is going to go through filtration, while the venous end is going to go through reabsorption. Okay? I put all of these in words to read them over. Now, what I just said is in this graph. So all the Starling forces stay the same except for the hydrostatic capillary pressure. As you can see, it decreases. Now, because it decreases, the difference between the hydrostatic pressures also decrease, as you can see. Everything else stays the same. Everything. See the oncotic capillary pressure, oncotic interstitial fluid pressure, and the capillary interstitial fluid pressure. Now, the difference in the hydrostatic pressures here in the beginning is higher than the difference in the oncotic pressures. This is where filtration is filtered. And once it's reduced, this is where reabsorption happens. Okay, so now we're done with these systemic capillaries. Moving on to the glomerular capillaries, which are the important parts. This is what you need to know for your exam. Now, same thing, Starling forces, there's 4 of them. Instead of the interstitial fluid, we're going to talk about the Bowman space. Now, to begin with, the oncotic pressure of the Bowman space is nearly 0. Why is that? We previously mentioned that the basement membrane and the filtration membrane in general is going to prevent plasma proteins from passing through. So in normal conditions, the oncotic pressure of the Bowman space should be 0 or should be nearly 0. And the hydrostatic pressure of the capillary is going to remain constant throughout the capillary, unlike the systemic capillaries. Why is that? Because glomerular capillaries have a higher radius. Now, if they have a higher radius, that means they have less resistance. So the resistance is very, very low. And because of that, pressure is not affected, the hydrostatic pressure, so it stays constant and high throughout the capillary. Now, same thing goes for the hydrostatic Bowman space pressure. It's going to stay the same because when fluid is filtered, it's going to go throughout the tubular system. It's going to continue. It's not going to stay in the Bowman space. And finally, the oncotic pressure of the capillary is going to increase as it goes along the capillary, unlike the systemic capillary where it stays the same. Now, if there's any disease such as a tumor or a stone that is in the kidney, that's in the tubular system, it's going to block the flow. For example, let's say a stone is right here. This is where the hydrostatic pressure of the Bowman space increases. This is abnormal. This is not supposed to happen. Again, we mentioned that this should be nearly zero. In case of a nephrotic syndrome or disease, the podocytes are damaged, which allow plasma proteins to pass through. So in a case like this is where the pressure increases, and that's abnormal. Again, it should be nearly zero. Now, we talked about systemic capillaries, and we said that reabsorption and filtration happens. In this case, in glomerular capillaries, reabsorption does not happen. It's filtration. Now, why does this happen? It's because the hydrostatic pressure of the capillary stays at a high level and is constant. So when the oncotic pressure increases, it stops at a level where it cannot exceed the hydrostatic pressure. So this opposes filtration. However, it only stops it. It does not cause reabsorption to happen. And finally, final point, you can notice the renal plasma flow is 600. The filtration is 120. This is an extremely high percentage. This is 20% of the total blood flow is being filtered. What we said in systemic capillaries, it's 0.5%. So this is 40 times higher than what we see in systemic capillaries. And this proves that glomerular capillaries are very important for filtration. Again, all I said is written here in text. Hydrostatic pressures, they push fluid out to the opposite direction. Oncotic pressures pull fluid. Oncotic pressure is zero. And we talked about nephrotic disease. This is a recap, just a comparison. There's no reabsorption in glomerular capillaries. However, in systemic, there is. The hydrostatic pressure of the capillary is unchanged due to the low resistance and high radius of the capillary. However, in here, the hydrostatic pressure of the capillary decreases because there's high resistance. And finally, GFR is much more higher in glomerular capillaries than in other systemic capillaries. Okay, now we're going to start with questions. Type your answer in the chat, okay? So why does filtration occur throughout the entire length of the glomerular capillary, unlike the systemic capillaries? Oh, okay, so the answer is B. Now let's read the choices and break them down. So we talked about how the glomerular hydrostatic pressure stays high along the capillary here. This is the hydrostatic pressure, and this is what causes reabsorption to not happen and just allow filtration to be there throughout the capillary. A is incorrect because oncotic pressure of the Bowman space is zero. We talked about it. It shouldn't be any number at all because plasma proteins don't even filter through the membrane. And here, glomerular oncotic pressure remains constant. That's incorrect because it rises. Bowman space pressure surpasses glomerular pressure. That's incorrect because in this case, filtration doesn't happen at all. And finally, Kf is lower in glomerular capillaries. That is incorrect. It is higher here. Okay, now what if renal plasma flow increases? See, this is the normal renal plasma flow, and this is the increased renal plasma flow. You see, it's doubled, and now the GFR also doubles. It was to 120, now it's to 140. Now, because there is double amount of plasma that needs to be filtered, it's going to need the double of the time that it usually needs to filter this. So it gives a slower rise with the oncotic pressure because there's more time to filter the blood, so it takes longer to reach the level it usually reaches. So two changes that happen with increased renal plasma flow: higher GFR and a slower rise in oncotic pressure in the capillary. Okay, so there's a lot of numbers in here. We're going to talk about everything. So here you can see the renal artery pressure and the peritubular capillary pressure. Those two pressures are unchanged. Unlike here, the glomerular capillary pressure. See, numbers are being changed, and we're going to explore how that happens. Now, this model is theoretical, does not happen in real life, but it helps explain a concept. Now, those two numbers, those two columns, are the resistance of the afferent and efferent arteriole. Now, the total resistance is 2 for both arterioles. We're not going to change that. We're only going to change the distribution of the resistance. Now, the pressure in the glomerular capillary is 60. What if we removed a half from the efferent and added the half to the afferent arteriole? So now there's more resistance in here. Now, the more resistance causes the lumen of the capillary the arteriole, to be narrower, unlike here. You can see the difference. Now, this difference in the size of the lumen is going to have a greater drop in pressure and cause the pressure in the glomerular capillary to be less. Now here, the opposite happens. We decrease the pressure here and we increase the pressure here. If we decrease the pressure in the afferent arteriole, you're going to see that the lumen is larger and more blood flows through the arteriole, and there's a higher pressure in the capillary. Now we're going to apply this to this equation over here. The flow rate equals the change in pressure over resistance. Now the change in pressure, they're referring to the pressure between the renal artery and the peritubular capillary pressures. They're unchanged, so the difference is still the same. It's 80. Now what we changed is the resistance. So resistance increases, flow rate decreases. They're inversely proportional. Because here the resistance increases, lumen is less, less blood flow is going in, so the flow rate decreases. The opposite happens here. Okay, this is it for the slide. Moving on to the next. Oh, sorry. No, you don't need to know the diseases. You don't need to know the pathology or anything. Dr. Shams only put these in the slides just for you to apply the concepts that you've learned. Okay, so you don't need to know the diseases at all. Now, what if we constrict one of the two arterioles? We're going to start with the afferent arterioles, okay? If we constrict the afferent arteriole, as we said, less blood is going through, so the renal plasma flow is less. Plus, the hydrostatic pressure is also going to decrease. Why? Because the hydrostatic pressure is dependent on the fluid. Now, if less fluid is going in, so less hydrostatic pressure is going to be in the glomerular capillary. Now, both decrease. Oh, hold on. Now, when they both decrease GFR. And those two variables, collectively, they still are going to decrease GFR in the end. So, constricting the afferent arteriole gives two effects: decrease in RPF, decrease in hydrostatic pressure of the glomerular capillary, which ultimately decreases the GFR. The case is different when we only constrict the efferent arteriole. Again, renal plasma flow decreases. Why? Because the blood is going to decrease again. Less blood flow is going to go through. However, hydrostatic pressure is going to increase for the capillary because more blood is being pulled up in the glomerular capillary. So, when renal plasma flow decreases, GFR decreases. Because do you remember here, when it increases, GFR increases. So the opposite is true. When it decreases, GFR also decreases. But because of the increase in hydrostatic pressure, the GFR increases because, again, we said the hydrostatic pressures, they push fluid out. So it pushes more fluid out into the Bowman space. So this increases the filtration rate. Okay, so if one decreases, one increases. What is the final effect? Now, if there's only moderate constriction, GFR increases. Why? Because the hydrostatic pressure dominates over the renal plasma flow. So its effect is, it's more effective than the renal plasma flow. And it's the opposite when there's severe constriction. This is where GFR decreases because the renal plasma flow has a more effect than the hydrostatic pressure. So you can, oh, hold on. So this is the line, okay? In moderate constriction, the GFR increases and it comes to a point where it remains unchanged. But then when you constrict the arteriole more and more, it's going to decrease the GFR. So the GFR can remain unchanged or can increase or decrease in the constriction of the efferent arteriole, okay? But for the afferent arteriole, GFR decreases in all scenarios. Okay. There's a lot of information in here, but we've mentioned most of it in the previous slides. So this is what affects the GFR. We got the filtration constant and the Starling forces. Now let's start with the filtration constant. We mentioned that it's it's responsible for GFR. How? By we decide how much it affects it with the permeability of the capillary and its surface area. If they increase, if both of them increase, the constant increases and GFR increases, and the opposite is true. So if we have a disease like glomerulonephritis where the capillary is inflamed, the lumen decreases and the capillaries itself is going to be damaged. So when they're damaged, there's less surface area and less filtration, and that will affect the GFR. Moving on to the hydrostatic pressure of the capillaries. Now, we mentioned this in the previous slide. If the afferent arteriole is constricted, the hydrostatic pressure decreases. If it's the efferent arteriole which is constricted, the hydrostatic pressure increases. And we talked about its effects in this previous slide. Moving on to the hydrostatic pressure of the Bowman space. I also mentioned this previously. If there is congestion of urine and urinary system, such as presence of a stone or a tumor, the hydrostatic pressure of the Bowman space increases because there's a buildup of fluid in that space. Now, this will cause the GFR to decrease. Why? Because if the hydrostatic pressure wants to push the fluid back into the capillary, it does not favor the filtration. So this is where GFR decreases. Moving on to the oncotic pressures. Now, the oncotic pressure of the capillary increases by dehydration. Why is that? Now, with dehydration, the hydrostatic pressure decreases. So there's a more concentration of the on the plasma proteins in the capillary itself. So this increases the oncotic pressure of the capillary, thus decreasing the GFR because again, oncotic pressure helps pull fluid. So it does not like the fluid to be filtered through the membrane. And the oncotic pressure of the capillary decreases by hunger. Why is that? Because with hunger, you're going to produce less proteins by the liver. So that is going to increase the oncotic pressure. Plus, with severe hunger, the body is going to start breaking down the proteins that are in the blood for other metabolites and to produce ATPs. So all of this will decrease the oncotic pressure. And this will increase GFR, opposite of the previous case. Now, the oncotic pressure of the Bowman space is normally nearly zero. However, it increases by diseases of the glomeruli. And finally, because again, if podocytes are damaged or endothelial cells are damaged or basement membrane, it's going to allow more proteins to be filtered through the membrane. So that's going to increase the oncotic pressure in the Bowman space. And finally, the renal plasma flow, it's an easy concept. If it increases, GFR increases. If it decreases, GFR decreases. Now we're going to move on to the questions. Which change will increase GFR? No, it's not B. Do you want to do a second try before I explain all the choices? Okay. So why is it not B? Let's start with B. Increased hydrostatic pressure of the Bowman space. This is going to decrease GFR. Why? Let me go back a few slides. Here, over here, hold on. Yeah. We said that hydrostatic pressures, they push fluid. So the hydrostatic pressure of Bowman space will push fluid into the capillary, while the hydrostatic pressure of the capillary pushes fluid into the Bowman space. It's an opposite relation, unlike the oncotic pressures where they pull fluid into their location. Now we go back to the question. So if we increase the hydrostatic pressure in the Bowman space, it's going to decrease GFR because it does not favor the fluid to be filtered into the Bowman space. And if we increase oncotic pressure in the glomerular capillary, it's going to cause less fluid to be filtered because here, the increase in the oncotic pressure of the capillary is going to decrease GFR, not increase it. If we constrict the afferent arteriole, what did we say? It decreases renal plasma flow and it decreases the hydrostatic pressure in the capillary. And they both decrease GFR, so that's incorrect. However, in option D, it is the opposite of question C. It dilates the afferent arteriole. So this causes more blood to flow into the capillary, and it's going to cause an increase in hydrostatic pressure, and they both increase GFR, so D is the correct option. And E is incorrect because it's severe constriction of the efferent arteriole. We said that the moderate constriction is going to increase GFR, but the severe one is going to decrease GFR. So if E was moderate constriction, it would be correct. The second question, if a patient has an obstruction in the ureter and it causes a backup of fluid, which Starling force increases? The correct answer is C. Oh, sorry, not C, B. Because what did we say over here? Usually fluid just flows through the tubular system when it's being filtered. But if there's a tumor, for instance, over here, the Bowman space is going to have a buildup of fluid. It's because the fluid is not going to be able to go back into the capillary, so it's just going to stay in the Bowman space. So there's a lot of fluid. That means hydrostatic pressure increases. So the correct answer is B. Okay, any obstruction in the ureter affects the hydrostatic pressure of the Bowman space. Everything is clear? Okay. C is incorrect because again, the fluid does not back up into the capillary, so the hydrostatic pressure of the capillary is unaffected by anything that's going on in the tubular system. Third and final question, which change occurs when renal plasma flow increases while other factors remain constant? Okay, so the correct answer is C. Faster rise, B, sorry, slower rise in the oncotic pressure of the glomerular capillary and an increase in GFR. Those are the two effects that are happening due to an increase in renal plasma flow, because here in the slide I mentioned those two effects. Here, as you can notice, there's a slower rise in oncotic pressure, unlike here, and GFR also increases because, for instance, if the amount of renal plasma flow is doubled, the GFR also doubles, okay? So a slower rise in the oncotic pressure of the glomerular capillary allows more blood to be filtered, okay? Any questions so far? Okay. Now, this graph shows you all the vessels, or most of the vessels that are involved in this system. We're going to focus on these three, okay? So this is the afferent arteriole, or they're also called vas afferents. And this is the efferent arteriole, which is also called the vas efferent, okay? Now you can notice the greatest drops in pressure is over here, those two arterioles. Now you can see here it remains constant in the glomerular capillary, the pressure remains constant, because we said that the resistance is very low in this capillary. So if the resistance is very low, the pressure is unaffected and will remain constant throughout this vessel. Now, why is there a lot of pressure drop in both of these the arterioles? It's because there is a large resistance in these arterioles. And if there's a high resistance, that means pressure is going to be affected. Now, let's say a patient has a pressure of 190, for instance. What happens? Oh, sorry. Hold on. Okay. So let's say that their pressure is over here, 190, 200, any number. Everything stays the same except for one thing. The greatest pressure drop is going to be here, in the afferent arteriole. The pressures throughout all of these vessels is going to remain constant. But in the case of hypertension, the greatest drop is going to be visualized over here, in the afferent arteriole, okay? Those are the only concepts that you need to know over here. Now, let's look at this graph. You could notice between, let's say, 90 to 180, the renal plasma flow and the GFR remains constant between those pressure values. Why is that? We have a mechanism that is auto-regulated by our body, and that is helped by the resistance. So the resistance changes with the pressure in the vessels, so that helps maintain a normal range of pressure. Now, the two mechanisms that are in or are a part of the autoregulation are the myogenic mechanisms and the tubuloglomerular feedback. Now, keep in mind, if the pressures are out of the range of this, so if they're less than 90 or more than 180, this is where the mechanisms start to fail. So first, let's start with myogenic mechanisms. The blood pressure increases, so what happens? The afferent arteriole is going to stretch. Now, this stretching is going to release calcium into the cell of the smooth muscles. Now, when calcium is released, what happens? It goes back to being constricted, because the vessel is going to try to bring the blood pressure back to normal. So when it constricts, it's going to lower the blood pressure. So this helps auto-regulate the blood pressure, not making it too high or too low. Now, this doesn't work, again, if the pressure goes out of this range. Okay? Now, the second mechanism, but before we get into it, I'm just going to go into quick anatomy. This is the renal corpuscle, and this is the distal tubule. Now, there is a renal tubular system. There's the proximal tubule, loop of Henle, and distal tubule. This is where it touches the renal corpuscle because remember, it's twisted. They're not all straight. They're going to overlap one over the other. So the macula densa in the distal tubule, there's specialized cells located in here which interacts with other cells. It interacts with the extraglomerular mesangial cells and the juxtaglomerular, or also called granular cells. Okay, those three, macula densa and the extraglomerular mesangial cells, they're all part of something that is called the juxtaglomerular apparatus, okay? Not important for this lecture. You covered it in histology, but just a quick reminder. Three components. So, what is the function of macula densa? It helps monitor the sodium and chloride levels in the tubular fluid. So if there's any rise or any decrease in amount, it helps regulate blood pressure and GFR by sensing those levels. And it also controls renin release from the juxtaglomerular cells over here. We're going to talk all about it in the next few slides. Don't worry, I know there's too many things, too many functions, but we're going to get into it. Now, we're going to start with the second autoregulatory mechanism, which is the tubulo-glomerular feedback. From its name, tubulo, so the distal tubule is going to sense any changes in the fluid, and the glomerulus is going to help change certain factors to help bring things back to normal. So, if GFR is increased, what happens? More things are being filtered, more ions are being filtered. So the macula densa is going to detect more sodium and chloride levels in the fluid. When it detects it, there's going to be more ATP and adenosine concentrations. And because of this increased concentration, the ATP is going to bind to the P2X receptors, and the the adenosine is going to bind to the adenosine receptors. So they're going to bind to their respective receptors in the smooth muscle of the afferent arteriole. And what this causes is an increase in calcium concentration. So this increase in calcium concentration, remember, it constricts the afferent arteriole, which brings blood pressure down. Or sorry, it does not bring blood pressure down. Yeah, it does, but indirectly. But it also decreases the blood flow in the glomerular capillaries, and decreasing GFR to help bring the substance concentration back to normal. Now, the vascular smooth muscle over here is going to electrically couple with the granular cells over here. What this causes, again, increased concentration level of calcium in the granular cells, which is also called the juxtaglomerular cells, and this will cause a decrease in renin release. Now, this decrease in renin release is going to cause the vasodilation of the other capillaries, not the glomerular capillaries, the other capillaries. This causes the vessels to widen and also helps in decreasing blood pressure. So this affects the glomerular capillary. This causes the effects in the other capillaries. Now, if GFR decreases, everything that I just mentioned it's going to happen in the opposite way. So this decreases, and so on. In addition to that, when GFR decreases, there's going to be an increased production of prostaglandins, which also help in increasing renin secretion, increasing blood pressure, and so on. Now, we talked about autoregulation. We're going to talk about the other mechanisms that help affect renal blood perfusion. Now, let's talk about the nervous system. If there's an increased moderate sympathetic activity, the effect dominates on the efferent arterioles. Here, the GFR remains unchanged because we said if the efferent constricts, it could either increase, decrease, or remain unchanged depending on the degree of constriction. So here, GFR remains unchanged. However, The filtration fraction increases from this equation. If filtration fraction increases, that means that the renal plasma flow decreases because the arteriole is constricted, so less plasma is going through. Now, if the sympathetic activity is severe, the effect is going to be more on the afferent arteriole. And we mentioned this multiple times. The constriction on afferent arteriole is going to decrease GFR. Now, in the case of hypovolemic shock, this is what happens. So there's very, very little renal blood perfusion in the kidney because the body is more concentrated on delivering the blood into more important structures such as the brain or the heart. So this puts kidney in shock. And there are multiple hormones that help influence the renal blood perfusion. The renin, angiotensin, aldosterone mechanism, we'll get into it in the next slide. The atrial natriuretic peptide helps dilate afferent arterioles. We mentioned it increases renal plasma flow, also renal blood flow, and GFR. And prostaglandins and nitric oxide, they help dilate arterioles, and that increases renal blood flow and GFR. Now, I previously mentioned prostaglandins help constrict arterioles. However, there are multiple types of prostaglandins. So there are those who vasoconstrict, there are those who vasodilate. So don't confuse yourself with that part. So now this is the renin, angiotensin, aldosterone pathway. So there are multiple things that trigger renin release, such as the sympathetic nervous system, or as we mentioned previously, if GFR decreases over here. If GFR decreases, renin secretion increases. So the kidney produces more renin or releases more renin, and the liver produces angiotensinogen. Now the renin converts it to angiotensin 1. And the angiotensin converting enzyme that is produced by the lungs is going to convert angiotensin 1 to angiotensin 2. Now, angiotensin 2 has many, many effects. First, it's going to increase thirst, so that's going to cause us to take more water, feeling the urge to drink more water. So that helps increase blood volume and increase blood pressure. Decrease blood pressure, sorry. And it also helps in constricting the blood vessels. It also helps in increasing the aldosterone secretion in the adrenal glands. And it also helps the reabsorption of sodium and water back into the peritubular capillaries, for instance. Now, all of this is written over here. So if there is any reduction in renal perfusion or there is decreased levels of sodium, it will stimulate renin secretion. Now, renin is going to convert angiotensinogen to angiotensin 1, and that will also help increase angiotensin 2 production levels. Now, this will produce 4 effects that I've mentioned already. It increases antidiuretic hormone levels, which causes the reabsorption of water into the kidney, in the kidneys, sorry. So it goes back into the vascular system. It also helps in vasoconstricting vessels. This causes the increase in total peripheral resistance. And it helps the increased release in aldosterone, which helps the sodium and water reabsorption in kidney. And finally, thirst. It helps produce the sensation of thirst. Now we're done with all the concepts of this lecture. We're going to solve the final 2 questions, I believe. So autoregulation of GFR between mean arterial pressure of 90 to 180 is achieved by which mechanism? I'm waiting for your answers in the chats. So We said autoregulation is by two mechanisms. We said the myogenic mechanism and the tubular-glomerular feedback. So which one of those two is mentioned in these choices? It's B, myogenic response of the afferent arteriole. Because again, let's recap. Blood pressure increases, so that causes the stretching of the afferent arteriole, which will ultimately causes the rise in calcium levels in the cells, in the smooth muscle cells of the vessel. And finally, this will cause the constriction of the arteriole to help bring back blood pressure back to normal. Now, AMP release causes which physiological change? The answer is C. Dilation of afferent arteriole, which causes the increase in GFR. Over here. It is written. Okay? And finally, which of the following would increase renin release? No, A is incorrect. The correct answer is D. I'll tell you why. Now, if we go back to the tubulo-glomerular feedback, if there is an increase in sodium chloride levels, it's going to help decrease renin because this reflects the high GFR. So we're trying to decrease renin secretion in order to bring back blood pressure back to normal. Here. See, we asked for what would increase renin release. The increased levels of sodium and chloride in the macula densa will decrease the levels, the secretion levels of renin. B is incorrect because, again, increased ATP and adenosine levels at macula densa will help bind to receptors in the vascular smooth muscles, and that will, after a few steps, it will cause the decreased level in renin release. Now, the increased intracellular calcium levels, again, decrease in renin. Now, decreased sodium delivery to macula densa, it's the opposite of A, so D is correct. Okay, this is it. If you have any questions, feel free to ask. But this is the end of the session. You can scan the QR code to solve practice questions and give feedback. If you have any questions, feel free to ask. If not, this is the end of the session. You have my email and number. If you want to contact me later on for any more questions, feel free to do so. Thank you.